The overall goal of this functional genomic technology is to identify the precise genomic locations that a given protein occupies in living cells.
This method can help answer key questions in the fields of gene regulation and epigenetics, such as how the transcription machinery assembles and operates in the context of chromatin. The main advantage of this technique is that it reveals the exact genomic location for protein-DNA interactions at near base pair resolution with exceptionally low background.
Generally, individuals new to this method will struggle because the protocol is complex and requires attention to detail throughout.
To sonicate nuclear lysates, begin by placing resonance adapters in 15 ml polystyrene tubes containing nuclear extracts. Sonicate the lysates in an ice-cold water bath at medium power for 30 seconds on and 30 seconds off, for a total of 2 15-minute sessions. To check sonication results, transfer 2 10 microliter samples from each tube into 1.5 milliliter tubes. Then reverse cross-links by combining the first 10 microliter sample with 10 microliters of tRNase A and 0.2 microliters of proteinase K. Incubate the tube at 37 degrees Celsius for 30 minutes. Then add 4 microliters of 6X-xylene DNA dye. To the second 10 microliter sample, preserve crosslinks by adding 2 microliters of 6X-xylene DNA dye. Run both samples on a 1.5% agarose gel at 140 volts for approximately 30 to 45 minutes, until the bromophenol dye in the latter is 3/4 of the way down the gel. If sonication was successful,